ขอต้อนรับทุกท่านเข้าสู่กิจกรรมในช่วงถัดไปนะครับซึ่งจะเป็นการกล่าวแนะนำความเป็นมาของโครงการ JAPAN-ASEAN BRIDGES Event Series โดยคุณ Uwe Morawetz ประธานกรรมการและผู้ก่อตั้งมูลนิธิสันติภาพนานาชาติครับ Welcome all distinguished and honored guests and participants to join the next session. We will begin with the introduction and background of the JAPAN-ASEAN BRIDGES Event Series, which will be presented by Mr. Uwe Morawetz, a founding chairman of the International Peace Foundation. Mr. Uwe, you may begin when you're ready.
Welcome to the Japan-ASEAN Bridges Event Series, facilitated by the Vienna-based International Peace Foundation. The events are hosted in cooperation with the various local partners, including some of the country's major universities, and I would like to thank Ramkhamhaeng University for hosting our event today. Commemorating the 50th anniversary of official relations between Japan and the ASEAN region, Bridges will be continuously held in Thailand and Japan until March, involving the participation of Nobel Laureates for physics, chemistry, medicine, and economics. The Japan-ASEAN Bridges series follows the series of over 800 Bridges events, which the International Peace Foundation has facilitated since 2003 to support education in the ASEAN region. The pluralistic program of Bridges highlights the International Peace Foundation's intercultural and transdisciplinary approach towards peace. The foundation doesn't take sides, but acts as a mediator by creating an independent platform for dialogue where representatives of science, politics, economy, culture, religion, the media, and youth can meet, share their viewpoints, listen to each other, and find mutual ways of understanding and cooperation. Therefore, the foundation itself is a bridge and the facilitator between different language groups in our divided societies, where politicians speak another language than artists, and business and religious leaders another one than scientists. In a highly interdependent world, problems cannot be solved only by either one of these language groups only, but by working together. The aim of Bridges is to facilitate and strengthen dialogue and communication between societies in Asia with their multiple cultures and faiths, as well as with peoples in other parts of the world, to promote understanding and trust. The events aim at building bridges with local universities in Asia to establish long-term relationships with Nobel laureates in all fields, which result in common research programs and other forms of collaboration. By enhancing science, technology, and education as a basis for peace and development, the events may lead to a better cooperation for the advancement of peace, freedom, and security in the region, with the active involvement of the young generation, ASEAN's key to the future. This is why Bridges is not designed as a one-time event, but as a continuous process of synergies of now over 2 decades to make the series of events a sustainable success for Thailand and for Asia as a whole. I am grateful to our honorary chairman, Anand Panyarachun, and our partners and sponsors who have enabled us to make the idea of Bridges a reality. I would like to thank everyone present today for taking part in this program. May it help us to facilitate a new culture of peace through dialogue, transcending its definition as merely the absence of war or armed conflict into a new understanding of what the basis for peace is: education. In this spirit, we welcome today the 1993 Nobel Laureate for Medicine, Dr. Sir Richard Roberts, who has again agreed to come to Thailand to support the events. We all look forward to his important keynote speech and to his important contribution to build bridges. A warm welcome, Sir Richard.
ขอขอบพระคุณคุณอูเว่ มอราเวสนะครับประธานกรรมการและผู้ก่อตั้งมูลนิธิสันติภาพนานาชาติที่ได้ให้ข้อมูลความเป็นมาของโครงการ Japan-ASEAN BRIDGES Event Series ครับ We would like to thank you very much Mr. Morawetz for providing us the introduction and background of the Japan-ASEAN BRIDGES Event Series แล้วก็มาถึงไฮไลท์ของกิจกรรมในวันนี้นะครับนั่นคือปาฐกถาพิเศษโดยศาสตราจารย์ดรเซอร์ริชาร์ดโรเบิร์ตนะครับนักวิทยาศาสตร์รางวัลโนเบลสาขาการแพทย์ปีพุทธศักราช 2536 นะครับปัจจุบันท่านเป็นประธานบริหารแผนกวิทยาศาสตร์ของ New England Biolab นะครับในรัฐแมสซาชูเซตส์ประเทศสหรัฐอเมริกานะครับท่านสำเร็จการศึกษาในระดับปริญญาตรีสาขาเคมีในปีพุทธศักราช 2508 และปริญญาเอกสาขาเคมีอินทรีย์ในปีพุทธศักราช 2511 จากมหาวิทยาลัยเชฟฟิลด์ประเทศอังกฤษครับท่านได้ทำวิจัยหลังปริญญาเอกในห้องปฏิบัติการของศาสตราจารย์สตรอมมิงเกอร์ที่ฮาร์วาร์ดนะครับประเทศสหรัฐอเมริกาโดยศึกษา tRNA นะครับที่เกี่ยวข้องกับการสังเคราะห์ทางชีวภาพของผนังเซลล์แบคทีเรียครับตั้งแต่ปีพุทธศักราช 2515 ถึง 2535 ท่านย้ายไปทำวิจัยที่ห้องปฏิบัติการโคสปริงฮาร์เบอร์นะครับและได้ดำรงตำแหน่งผู้ช่วยผู้อำนวยการฝ่ายวิจัยภายใต้การดูแลของดร.วัตสันโดยทำวิจัยเกี่ยวกับเอนไซม์ตัดแบบเฉพาะเจาะจงประเภทที่2นะครับแล้วก็ที่เพิ่งค้นพบใหม่ในปีพุทธศักราช 2515 และในอีกไม่กี่ปีถัดมานะครับท่านได้ค้นพบเอนไซม์เพิ่มขึ้นมากกว่า 100 ชนิดและยังได้ระบุลำดับของนิวคลีโอไทด์ของจีโนมอะดีโนไวรัส 2 ได้ถึงเกือบ 36,000 หน่วยและท่านยังได้เขียนโปรแกรมสำหรับการจัดเรียงและการวิเคราะห์ลำดับนิวคลีโอไทด์อีกด้วยนะครับท่านได้รับรางวัลโนเบลสาขาการแพทย์ในปีพุทธศักราช 2536 นะครับจากการค้นพบยีนที่ทำหน้าที่แยกตัดแต่งและเชื่อม mRNA การค้นพบของท่านนะครับได้เปลี่ยนวิธีคิดของนักชีววิทยาที่เกี่ยวกับยีนไปอย่างสิ้นเชิงและองค์ความรู้ดังกล่าวนี้นะครับทำให้เกิดความก้าวหน้าด้านการวิจัยอื่นๆนะครับอีกหลายสาขารวมทั้งการวิจัยเกี่ยวกับโรคมะเร็งด้วยและในวันนี้นะครับท่านจะปาฐกถาในหัวข้อเรื่องการเผยแผ่สันติภาพด้วยวิทยาศาสตร์และการพาณิชย์ในประเทศเศรษฐกิจเกิดใหม่ครับ
Now we are approaching the highlight of the day, which is the keynote lecture by Professor Dr. Sir Richard Roberts, a Nobel Laureate for Medicine in 1993. Currently, he is the Chief Scientific Officer at New England Biolabs in Massachusetts, the USA. He obtained a Bachelor of Science in Chemistry in 1965 and a Doctor of Philosophy in Organic Chemistry in 1968 from the University of Sheffield. His postdoctoral research was carried out in Professor Strominger's laboratory at Harvard University in the US, where he studied the tRNAs that are involved in the biosynthesis of bacterial cell walls. From 1972 to 1992, he worked at the Cold Spring Harbor Laboratory, reaching the position of Assistant Director for Research under Dr. Watson's supervision. He began work on the newly discovered type 2 restriction enzymes in 1972, and in the next few years, more than 100 such enzymes were discovered. Professor Roberts' laboratory sequenced almost 36,000 nucleotides adenovirus 2 genome and wrote some of the first programs for sequence assembly and analysis. Professor Roberts was awarded the Nobel Prize for Medicine for his discoveries of split genes an mRNA splicing. His discoveries completely changed the way biologists thought about genes and led to decisive progress in many fields, including cancer research. Today, he's going to deliver his keynote lecture on spreading peace through science and commerce in emerging economies. Professor Roberts, the stage is yours. Please.
Okay, well, thank you very much. It's a pleasure to be here. I'm going to try and talk today about a subject that is really on everybody's minds at the moment, and that is peace. I think we look around, we see the news, everywhere we look, we discover that there are people fighting one another. And for me, as a scientist, I find this completely reprehensible. Maybe this doesn't work, okay. Well, the slides don't seem to want to advance. Can someone help here? Really is there someone who can help advance the slides? Because none of this is working. This doesn't work and this does. Technology strikes again.
Okay, I think you may use this one.
Okay, I'll do it from this. Okay.
Thank you.
Okay. So, really, one of the messages that I'm hoping will come through very clearly during the talk today is that science is essentially a very peaceful activity. Scientists, we disagree about things all the time. We have a hypothesis. Maybe it's right, maybe it's wrong. But in every case, we resolve these disputes peacefully. We don't find it necessary to go and shoot someone who has a different opinion from ours. And that means that if you're a scientist, no matter where you go in the world, if you meet another scientist, you have a friend. And I view all of you here in this room who are scientists, you are all my friends, because science unites us in ways that other things often don't. And I would like to make the point that scientists also make extremely good diplomats. During the Cold War in Russia, I spent a lot of time going over, visiting, along with many of my colleagues, and also people from the arts. We went over, and this was a constant source of news, of real news, for the scientists and for the people that we met when we went to Russia. We were able to let the Russians know, the Russian people, what was going on in the rest of the world when their leaders were trying to tell them otherwise. Similarly, when China first opened up, a lot of Chinese scientists came over to the US. I had two who came to my lab at Cold Spring Harbor almost immediately after China opened up. And following Chinese scientists coming and learning from us in the US and the UK and elsewhere, we also went over and visited them over in China. And again, these were incredibly peaceful activities, because scientists are peaceful. In Cuba, there was tremendous antipathy between the Cuban government and the American government. Don Combe, who founded the company that I now work for, he used to go to Cuba regularly and take reagents, take supplies, scientific supplies that they could use over there. And later on, I too went over and spent time in China in Cuba trying to establish good relations with the Cubans, and I still have those today. And throughout the developing world, I and many of my fellow scientists travel regularly, trying to establish contacts, trying to help them to do well. Now, science leads to discoveries. And as soon as you make a discovery, you often find that there is the possibility for commerce. There is the possibility to take advantage of that discovery, to make a company, to do something that would be good for science or society or wherever. In fact, for the whole world. Science really opens up discoveries. And I will talk today about three things. The first of it is restriction enzymes. These were the enzymes that cut up DNA that formed the basis upon which the biotechnology industry was built. It also formed the basis upon which the company I now work for was formed. And I will tell you about that. I'll also tell you about the discovery of RNA splicing, for which I won the Nobel Prize. This discovery had implications immediately for basic research. Didn't have any commercial applications for the longest time. The discovery was made in 1977, but it wasn't until about 5-6 years ago that the first commercial, actually medical, application was found. And finally, I'll tell you about DNA methylation, which is something I work on today, for which we made a discovery a couple of years ago, for which there is now a clinical trial underway to try to take advantage of that in the medical arena. But I want to start by talking about New England Biolabs, the company that I got associated with. I got offered a job following my postdoc with Jack Strominger, shown here on the left. I learned how to sequence RNA. I learned how to do some basic molecular biology for sequencing using a method developed by Fred Sanger, shown on the right, who was one of the most charming, most delightful Nobel Prize winners I've ever met. He actually won the Nobel Prize twice, once for developing methods for sequencing proteins, once for developing methods for sequencing DNA, and he could have had a third because he was the person who developed methods for sequencing RNA, which I learned during a visit in his laboratory. It turned out, by chance, that I was the first person in the Boston area at the time, I was a postdoc at Harvard, to actually start using Fred Sanger's methods and to enable them to be used in the Boston area, at Harvard in particular. And a lot of people came to my lab to learn how to do it. And so I actually made an awful lot of friends in those days because even though scientists can disagree, when you teach them something, when you help them, they become even more friendly than usual. So during that time, one gentleman, one assistant professor at Harvard, a man called Mark Potashny, who had figured out how bacteriophage lambda did control transcription and controlled it. He discovered the repressor and figured out how the repressor worked. He came to me one day and he said, you know, Jim Watson is going to come and see you and offer you a job at Cold Spring Harbor Laboratory. Well, I waited and no sign of Jim Watson. And eventually Mark came up to me and he said, Jim doesn't know who you are. Perhaps you could go and introduce yourself. And so I went over to his office, and I did. And I must have been in his office 10 minutes maximum, maybe even less. And the first thing he did was to offer me a job at Cold Spring Harbor to sequence SV40 DNA. SV40 DNA was one of these very small eukaryotic viruses that actually was able to cause cancer. And everybody at the time was interested in discovering the cause of cancer, and in particular, how we could prevent it. So I went down to Cold Spring Harbor, but when I got there, I discovered there were already 2 other groups who were sequencing SV40. And I don't like competition if it's unnecessary, which it was in this case. We didn't need a 3rd group to be sequencing it, and because these 2 groups were using exactly the methods I would have used. And so when I got there, I remembered that recently I'd been to a lecture by the man shown in the top left-hand corner of this slide, Dan Nathans. And he had given a talk at Harvard showing how with SV40 DNA, you could cut it with the very first restriction enzyme ever discovered called endonuclease R, and he showed you could cut it into small fragments. Now, one of the reasons that RNA sequencing preceded DNA sequencing was because there were plenty of small RNAs to practice on, but no small DNAs. And it occurred to me that if we could make some small DNAs with this restriction enzyme, maybe it would be possible to develop some new methods for sequencing DNA. And maybe there were more of these restriction enzymes out there, and that they could all be used to help develop methods to sequence DNA. Now, when I got to Cold Spring Harbor, there were 4 enzymes that were known, and I immediately purified them, and then went looking for more. And it turned out, by chance, everywhere we looked, there were more of these restriction enzymes. And because this was the early days, and because they have quite varied recognition specificities, they were all new. They all recognized different sequences. And so I soon reached a point where there were some 35 or 40 of these enzymes were known. I think only 2 of them recognized a sequence that was already known. And everybody found out about them because I would talk to my friends about them, and they used to come to Cold Spring Harbor, and they would say, well, you know, do you have a restriction enzyme that will cut my DNA? And it turned out they had a pot of their DNA in their pocket, and they'd say, you know, here's the DNA, do you have something that will cut it? And this turned out to be incredibly important for the biotechnology industry, and it occurred to me that here was an opportunity to make a company. Cold Spring Harbor Laboratory could easily make a company that would sell these restriction enzymes that I discovered. And in this way, we could both empower molecular biologists to do science and also make some money and use the profits to support the research at Cold Spring Harbor. All of this was really very, very obvious to almost everybody except for Jim Watson. And Jim Watson said, oh, you can't make money starting a company. Plus, this was back in 1974, 73, 74. Said, plus, it's really dirty to be associated with industry, and he didn't think this was appropriate. Well, well, I knew that, in fact, there were more restriction enzymes. There was a possibility of a company. And so I looked around, and I soon found that there was a company called at the time Biolabs that was formed in Beverly, Massachusetts by a man called Don Combe. And Don Combe had started a company, and he was selling reagents to research scientists, but not restriction enzymes. But he had thought that perhaps one of these restriction enzymes he would start to sell. Well, he did that in the sense that he contacted a distributor, Miles Laboratory at the time, and was going to set up an arrangement with them so that they would sell the enzyme he had made. I called him up and I said, look, we found at the time a large number of restriction enzymes. We ended up actually about 30 at the time. And I said, why don't you just make the ones, I can tell you which ones everybody wants because they all come to my lab to get free samples. I can tell you what they want. So why don't you start a company and sell it yourself? And that was the start of New England Biolabs. In 1975, New England Biolabs became registered as a company. We started to sell restriction enzymes to everybody who wanted them, and we would sell directly to the consumer, not go through a distributor. And this was unbelievably successful. I really can't tell you how shocked I was. In the first year of making these enzymes, this was 1975, we made $200,000 worth of sales. Huge amount of money just selling directly to consumers, something that we knew everybody wanted. And I've always looked upon this as a very good example of when you make a discovery and you realize it's something that people can use, there is always the opportunity for commerce. 1974 was particularly memorable. There was a meeting held in Ghent, in Belgium, and at the time, there were something like maybe 30, 35 restriction enzymes known, and I made the first compilation of these, as sort of a little database of what was known, and I've maintained that ever since. It's called Rebase, Restriction Enzyme Database, and I've maintained it, I've run it ever since, and now we have hundreds of thousands of entries in it. So this was the business model for Cold Spring Harbor, but it turned out that this business model was exactly the one that Don Combe had in mind. He had been an associate professor at Harvard and had not been given tenure. And so he decided that he didn't like Harvard very much and he didn't like the way academics treated people. And so he was going to start a company, but he was going to use the profits to support research. So instead of applying for NIH grants or NSF grants, he would just use the profits from this company to support research, and that is what's been done. Okay, this is our double helix related to New England Biolabs. And here's a picture of Don Combe, somewhat late in life. He died 3 years ago. And he ran the company much in the way that anybody who was an academic would think was a good way to run a company. And in fact, it was pretty much any way that a businessman would tell you not to run a company. So for instance, when we first started cloning the genes for restriction enzymes, we could make them very cheaply. Something that maybe used to cost us $1,000 to make, we could now make for $100. And so what did we do? We lowered the price. We dropped the price tenfold to just let everybody know we could do it. Our business competitors sent us hate mail. They said, "What's the matter with you? Don't you know how to run a business? When you figure out how to make something more cheaply, you pocket the profits. Go and buy a yacht or a home or whatever, but you don't lower the price." However, it turned out that as soon as we lowered the price, we sold 10 times more enzyme. And in fact, for us, it turned out to be very profitable to lower the price because we increased sales. And we also were cognizant of the fact that the cost savings going out to researchers around the world also made it possible for them to do experiments that they otherwise could not have done. And it's been a constant theme throughout New England Biolabs, throughout our lifetime, that we want to enable molecular biologists to do research. We want to make it easy, cheap for them to do it. This is the first catalog shown on the right-hand side, and it was the first catalog to have some scientific information in it besides just products. I had produced a list of all the known restriction enzymes, together with references as to where they came from, and that was included as part of this catalog. And if you now go to any company's catalog, whether it's in paper or on the web, you'll discover they're doing the same thing. They all copied us. They all thought this was a good idea. Now, from 1975 to 1980, we were in a basement in Beverly. It was actually a basement underneath a hairdressing saloon. And when I first went and met with Don Combe, there was Don, his wife, and one technician. And I was the fourth employee. Don actually offered me a share in the company, but I didn't want it. I wanted to do research, and I said, well, I'll just be your chief advisor. And so that was what I was. And I'm not an owner of the company, I'm just their chief scientific officer at the moment. Now, we quickly outgrew this basement in Beverly. We managed to get some space in an attic up above the original operation, but then we built a big building shown here at the top. This was the first building that we put up, and then we grew too big for that, and so in 2007 we moved into this current facility, and we've just added a 120,000-foot extension to this to accommodate the growth in the company. This is the people. Don Combe was the CEO. Then the company was 47 years old. It's now 49 years old. Jim Ellard took over as CEO, and most recently Sal Rossello. Both Jim and Sal are PhDs. The company is run by PhDs, by scientists. It's not run by businessmen. We have an award-winning catalog, a catalog that is now both available in paper form, and we still get a lot of requests for the paper form, but it's also available online. We have a wide range of products, not just restriction enzymes, but all sorts of other enzymes that are useful to the molecular biology company. And in this way, we enable research, we enable scientists who want to do molecular biology to do it in many ways that are possible. We're also a research institute. In Ipswich, Massachusetts, where the company is located, we have about 370 employees, of whom a little over 100 are doing research. All of it supported by the company. We have another 80 or so who are doing product development, improving products that we make, finding new uses for them, and making it into a proper company. We're also very philanthropic. We started something called the Ocean Genome Legacy Foundation a number of years ago, and they are collecting DNA from endangered marine species so that we don't lose the genetic information that is present in the oceans. Current research at the company involves restriction enzymes, DNA methyltransferases, which I'll tell you about later, genomics, filariasis. We made a decision back in 1984 that we were going to start some research that would help the developing world. We organized a meeting in which people talked about the major diseases that the World Health Organization was concerned about. Filariasis was one of them. This is a worm-based disease that causes problems in Africa, in South America. And during the course of a day's symposium, we discovered that there was very little money going into filariasis research, whereas all of the other things, tuberculosis, malaria, and so on, lots of money going into that. And so we picked filariasis and decided that we would fund some research to see what could be done. And in fact, we ended up collaborating with scientists in Africa. We brought them to New England Biolabs to train them. We sent them back to Africa. We sent money over there to help them pursue their research goals. We published a lot of work. And we actually developed a test for filariasis that is called a LAMP test, L-A-M-P. And it's a way of testing for the presence of DNA sequences from, in this case, a parasite, but in other cases, like coronavirus, you can do it without any fancy equipment. It's really easy to do. You can do it out in the field. You get an answer back within 30 minutes. And we developed a method for doing this for coronavirus so that we could protect the employees at our own company. We tested everybody 3 times a week. As soon as someone showed positive, they were sent home so that they wouldn't spread the virus around in the company. Stay home for 2 weeks, come back, test negative, they could come back to work, test positive, they stayed home. We tried to get this through the CDC. We tried to get the US government to say this was a good way of doing it. It was so much better than PCR, which is slow, it needs machines, it's expensive. But of course, you know, the federal agencies don't often listen to scientists in the way they should. And we have other things going on. Most recently, we have a big project in metagenomics. This is the extent to which we've been involved in humanitarian efforts. So we funded labs in China, in Cuba, in Nicaragua, Indonesia, Cameroon, Uganda, Portugal. And in each case, we would bring a scientist from that country over to biolabs, teach them what we do, and then send them back with some money so that they could start their own company, ideally, but at least could start doing research. We've always believed very heavily in philanthropy. Okay? We don't just want to use the money and buy fancy yachts and so on. We want to support research, whether our own or other people's. Now I want to switch now and talk a little bit about RNA splicing. So this was really an incredible discovery. I think it was one of these things at the time that we knew that it was going to be big, but I'm not sure we knew how big it was really going to be. What happened was a postdoc who had been a student at Harvard, who I'd met when I was a postdoc at Harvard, I invited him to come and work for me at Cold Spring Harbor. His name was Richard Gelinas. And he was given a very simple project, or we thought it was going to be a very simple project, which would be to find out if the signals in DNA of higher organisms, humans, human viruses, and so on, if the signal that told an RNA polymerase where to start making RNA was the same as it was in bacteria. And at the time, everybody thought that transcription in bacteria, this making of RNA in bacteria, would apply everywhere. I think it was Jacques Monod, a Nobel Prize winner, who famously said, what is true for the bacterium E. coli is true for an elephant. Well, that turned out not to be such a clever prediction. The idea for Richard's experiments were just to find what was at the 5' end of the messenger RNA, and we came up with a very clever method for finding that. Find where that sequence was on adenovirus 2 DNA, and then sequence in from the ends of the virus. Adeno is a linear virus. Sequence in from the ends and find out where the RNA sequence matched the DNA sequence. So we tried this very clever experiment, and everybody agreed it was a clever experiment, but it didn't give us the answer that we wanted. And it gave us a very strange answer. We were expecting to find a lot of different ends on the RNAs, but there was only one. And it was one of these cases where I thought, you know, Richard had messed up, and I said, go and do it again. And he did it again, and it still got the same result. And I said, let me show you how to do it, you know, because I'm pretty good. I did it and got the same result. And at that point, I believed it, of course. And so the question was, why were we getting this funny result? And I think there's an important lesson here, not just in science, but also in life. We had failed. We'd done an experiment and it didn't work. It was a failure. Now, a lot of people just stop and say, oh, it didn't work, and they go off and do something else. But I wanted to know why it didn't work and why it failed. Why did we have this failure? And the bottom line, to cut a long story short, is that nature was trying to tell us something. Nature was saying the hypothesis that transcription and making RNA in bacteria also works for all organisms just isn't true. And it took us quite a while to figure out what was going on, but eventually we did. This was the first paper that we found, and that is that all of the late messenger RNAs made in adenovirus all had exactly the same end on them. And the question was, how is this possible? It just doesn't make sense because they come from all over the DNA. How can they all have the same end? Well, we spent about a year doing experiments, trying every day to come up with something new that would tell us exactly what was going on. And then one day, early in March in 1977, Richard and I were talking, and he was coming up with a very complicated experiment he wanted to do. And all of a sudden it occurred to me, here is the experiment we should do. It's shown here on the right-hand side. And the idea with RNA, you can hybridize it to DNA. If you take a DNA strand, open it up, add RNA, you can form a DNA-RNA hybrid and displace the DNA strand. And we had discovered that the ends of the RNA didn't match the sequence right next to the main body of the messenger RNA. And I came up with this electron microscope experiment that would tell us, we thought, what was going on. Now, the only problem with that was that neither Richard nor I were electron microscopists, but fortunately, we had two friends who worked in the floor below us at Cold Spring Harbor, Tom Broker and Louise Chow, who were excellent electron microscopists. And so we went and we talked to them on a Saturday morning and said, you know, we have this idea for an experiment. Is it possible you could do it for us? And they looked and they said, well, it's never really been done before, but it looks as though it should be doable. So if you make the reagents, we will do the experiment. So Richard spent Sunday and Monday making the RNA and the reagents we needed for this. And on Tuesday morning, Louise Chow did the experiment. And the very first molecule she looked at looked exactly like the one that I'd drawn on the board on the Saturday. Except it was just a little bit different. Instead of there being some hybridization at just one point on the DNA, there were actually two. And it was already showing that the RNA that came from adenovirus was the final RNA that made it out into the ribosome to make protein, came from 3 different points on the DNA. And this was the discovery of RNA splicing. And basically, what RNA splicing is you make from DNA a very long piece of RNA, and then you cut the little bits and pieces that make sense, that are going to code for protein, and you cut and splice them in exactly the same way that a movie director makes a movie. The movie director shoots lots of small scenes, then they go to the editing room, they cut and paste the movie, cut the film, paste it together so that it looks like a continuous piece of action. That's how higher organisms make their RNA. They cut and paste little snippets of information to make something that makes sense. This was the paper that described that. It was called An Amazing Sequence Arrangement at the 5' ends of messenger RNA. And we had some interesting interactions over the word amazing. So I wanted to put in an adjective in the paper that was unusual, not something that you saw in every scientific paper. And my co-authors thought amazing was a little over the top, but nevertheless, eventually they agreed to allow me to use it and submit it. We had 3 reviewers. They each wrote back and they said, this is a great paper, big discovery, very important, but you know, amazing is not a word that you should use in the title of a paper. And so I called up the editor, Ben Lewin, and I talked to him for a little while about some of the other points that the reviewers had raised. And then I said, you know, when you first saw this, Ben, what did you think? He said, oh, it was amazing. I said, I rest my case. And so that is how the word amazing got into the title of a scientific paper. And as far as I know, it's the first time it was ever used in the title. It's been used since, but I've not found a paper that used it before then. Now, this led to the Nobel Prize in Physiology or Medicine in 1993, which I shared with Phil Sharp. Phil Sharp is someone that I had known well. He had been at Cold Spring Harbor when I first went there. He was a postdoc when I got my first job there. But he had left and gone to MIT and was independently working on transcription in adenovirus, and so made the discovery just a little after we did, but essentially simultaneously. And so we shared the prize. And I have to say, the one thing that, for me at least, comes from this, and also comes from talking to many other laureates, is that you cannot set your sights on wanting to be a Nobel Prize winner. I got the prize for making a discovery that was luck. We were doing an experiment, it failed, and it happened to fail because nature was telling us something big. And I think when you talk to Nobel laureates, almost all of them will say the same thing. They weren't trying to do something that would win a Nobel Prize, they were just trying to do good science. And if you do good science, you will always get interesting results. Sometimes they're big enough that you can get a prize for it, but other times they're not. But the sense of discovery is just wonderful. I do bioinformatics research. I open my computer every morning. I look at something new and I see something that no one's ever seen before. You cannot beat this feeling. This is just amazing. But on the other hand, if you do win a Nobel Prize, I thoroughly recommend it to everybody. Now I want to finish this talk by talking a little bit about DNA methylases. Now these are companions to the restriction enzymes because in order to protect the bacterial DNA against restriction, you have to modify the sequence. You put a methyl group onto the sequence. This is echo R1, GAA-TTC. You put a methyl group onto the A residue, and then the restriction enzyme can't cut it. And so then, when a phage comes in, its DNA is not modified. The restriction enzyme comes along and cuts it. And so you end up with a system in which you've got a methylase and a restriction enzyme. The methylase is protecting the host DNA against the action of the restriction enzyme. The phage is not such protected, gets cut, and so it's a kind of immunity for the bacterium. If we look at typical restriction systems, they come in 3 flavors, but the ones that I'm most interested in at the moment are the ones labeled 2, in which there is just 1 gene for a methylase, 1 gene for a restriction enzyme. Now, another piece of luck. In 1992, we started, well, actually in 91, we started working on trying to get a crystal structure for a DNA methylase. We wanted to know how it could recognize the DNA and methylate the specific base it was going to methylate. We tried several methylases with no success, and then in 1993, Jiadong Cheng, who was a postdoc crystallographer at Cold Spring Harbor Lab, we were collaborating with him, and he showed that when the methylase, M.HHA1, when it binds to DNA in order to put a methyl group on, it opens up the helix, flips the base that it's going to methylate right into a pocket in the enzyme. And this turned out to be the first time that anybody had seen base flipping, and we now know it is very common. Almost every enzyme that interacts with DNA to form modified residues uses this mechanism to do it. When RNA is being transcribed, with DNA rather, is being transcribed, very often there is base flipping that takes place. It's a common phenomenon. And I was lucky. I found out about this just 2 months before I had to go to Stockholm for the Nobel Prize ceremonies. And one of the things you have to do when you're there is give a talk. And most people talk about all the discoveries they made since they had made their original discovery and how they had used that original discovery to drive their research. I hadn't worked on it for many years, and this was the latest thing that we'd been doing, and we'd been doing it for a number of years. And so I was able to use this as my Nobel Prize speech and talk about a new discovery. And of course, I've always thought if you make a big discovery, it's not bad to be able to tell the Nobel Committee about it in person. Now, the one thing that we have discovered as a result of working in this field is that there are solitary methylases that don't have a restriction enzyme. They just have themselves. There is absolutely nothing that comes along. And so, what are these solitary methylases doing? They're not protecting the DNA against a restriction enzyme. What are they doing? Well, one of the best known is in E. coli, the DAM methylase, and it serves to mark which strand should be copied if mismatch repair is needed. In Colobacter, there's an enzyme, CCR1, that recognizes GANTC, and it is involved in the cell cycle, in monitoring the cell cycle. EcoG1 is a solitary methylase. It methylates only A residues, and we have no idea why it does it. But the most interesting one is this one from Clostridium difficile. Now, if any of you have ever been to hospital, you have to be aware the chance of picking up an infection from Clostridium difficile is very high. A lot of people go to hospital and pick up this infection. Most of the time, you get over it. Most of the time, they find an antibiotic that will help. But increasingly, this organism is becoming antibiotic resistant, and also it has a clever way of escaping from antibiotics. It forms bodies called spores, which basically are much like the spores that any fungus would form. It just stops growing, sits there, waits for the antibiotic to go away, and then it starts growing again. Well, it turns out, we discovered 2 years ago, that this enzyme from Clostridium difficile, which methylates GAA, GA5, and methylates the last A residue, if you knock out that methylase, C. difficile cannot sporulate anymore. It's controlling sporulation. And this means that one can think about an alternative treatment for C. difficile that doesn't just involve antibiotics, but involves something that would inhibit this methyltransferase. Several groups have now come up with an inhibitor, small molecule inhibitor, and it's undergoing clinical trials. And if we're lucky, then here will be another treatment to help solve the C. difficile problem. And C. difficile is particularly nasty because if you get an infection that you cannot get rid of, you end up with uncontrollable diarrhea, and people don't want to leave their house. They go home, they stay in their home, and they never leave their house because they don't know when they're going to be struck with diarrhea. So this would be really a wonderful treatment if, in fact, a compound can be found that will inhibit this. And the thing that I get excited about when I wake up every day is that there are many, many others of these solitary methyltransferases. What are they doing? I mean, there are literally hundreds of them. What are they doing? We don't know. I'm hoping either I will find out or perhaps some of you young people in this room will find out, because there's a very interesting biological problem going on here. Could have quite profound impacts on medicine, on food production, on all sorts of things. This is why I do science. This is why you should do science. And I think I've tried to give you some ideas. When you make discoveries in science, there are always commercial opportunities, commercial possibilities. Think about them. You don't have to do the company yourself. You can find a friend who would like to start a company. Talk to him or her and have them get on and do it. But there are almost always commercial possibilities when you make a discovery. So I wish you well. I wish you all make lots of discoveries. And if you do, email me and tell me. Thank you. Now, do you have... Oh.
ค่ะต้องขอบพระคุณศาสตราจารย์ดรเซอร์ริชาร์ดโรเบิร์ตสำหรับการบรรยายค่ะแล้วเรายังมีเวลาพอสำหรับคำถามสำหรับผู้เข้าร่วมงานทุกท่านนะคะโดยขอให้ท่านที่มีคำถามยกมือขึ้นนะคะหรือเดินมาที่ไมโครโฟนได้เลยค่ะและขอให้ท่านแนะนำตัวและหน่วยงานที่สังกัดด้วยค่ะ
We are thankful for your great talks, and we have some time for the Q&A
Sure.
sessions. Yeah.
I think there's a young lady over there. Okay, thanks.
Hi. My name is Apasala. I am a student of biotechnologies from Ramkhamhaeng University. Thank you for your lecture. It's inspired me so much. So my question will be like briefly because I have seen that an international peace foundation like cooperate with the conference this time here. I'm so exciting. And my question is that when you like cooperate with the other section with political specialists or human rights specialists or any others, how will you express yourself or your expression that your profession will help people being like better or enhance human rights? Yeah, because I have seen like many people are like youngster here, so yeah. So briefly, so I can tell like the younger people that yeah, you can help the world, you can save the day as well.
Yeah, you really can. I think it's very important that young people become activists and they don't just sit back and let the old folks walk all over them. But one thing that I discovered when I won the Nobel Prize was that whereas beforehand I'd always been quite active in stating how I felt about things, what I thought should be done when change was needed, in my opinion. And people would, sometimes listen, but there was never really any action that took place afterwards. But after I won the Nobel Prize, not only did people start to listen, but they also sometimes acted accordingly. And what I've been doing over the last more than 20 years now is actually organizing the Nobel laureates to make statements in support either of misinformation about science to correct any misinformation that comes about, but also to support humanitarian causes. And perhaps I can give you one example of the latter, which involved a little bit of science. And that was a number of years ago. There was an outbreak of HIV in the children's hospital in Benghazi in Libya. And there were some 400 children all got infected with HIV while they were in the hospital. And Colonel Gaddafi, who was head of Libya at the time, accused 5 Bulgarian nurses and 1 Palestinian student of deliberately spreading HIV to these children. But when scientists started to look at the records of what had gone on, it was clear, fairly obvious, that because they were reusing needles when they were giving vaccinations and so on, that it was because of this. Someone came into the hospital with HIV and then the nurses inadvertently spread it. They didn't do it deliberately, it was just standard medical practice in the hospital. Now, the nurses and student got put in jail, they were tortured. 3 of them confessed that they'd done it deliberately just to stop the torture, and they ended up being sentenced to death. Now, I read about this, when I first heard about it, it never occurred to me that anything would get this bad, but then I read an article in Nature saying that they were going to be put to death because of this. And I didn't think that was the right thing to do. And so I organized the Nobel laureates. We got more than 100 to sign a letter to Gaddafi saying that this had to stop, that they need to look at the science here and decide, what went on. It was an accident. It wasn't deliberate. And I went down and took this letter to the UN, the Libyan ambassador to the UN, and had a good conversation with him. Sent in the letter. He said he would deliver it to Gaddafi. After that, about 2 weeks later, I got a call from Gaddafi's son, Saif al-Islam, asking me if I would go to Tripoli and talk to him about this incident. I got on a plane, I went over there, met with him, and at the end of about an hour-long conversation, he said, "We're going to let them go. We know it wasn't deliberate." And so, thanks to the support of the Nobel Laureates, we were able to get justice for these nurses and the student. And I think this is one of the most dramatic examples that I've seen so far, but we continue to try to do things to get the laureates organized. But what I've learned as a result of this is that most of the time it's not the laureates themselves, it's rather the fact that because we speak out on something, politicians, others will act in ways that we had not anticipated, that we probably couldn't even act ourselves. And so our voices get amplified. And I think this is the message for young activists, is to find people that you can talk to so that the people who can do something really will try and do something.
Thank you very much. Any other questions from the floor? Okay, please.
Okay. My name is Nico, and I'm a master's student in Chulalongkorn University. And it's a big honor to see you here. I found one of your previous interviews with the editor-in-chief of the Nobel Prize in 2007. And you were said about that people who move from one field to another always come at the new field with a different underlying philosophy of how to do science, and that's a good thing. That my question is, should we as a researcher become a master of one or jack of all trades to be better researchers and answer the current problems? Thank you.
Okay, well, you're absolutely right. I did say that. I still believe it quite strongly. But part of the context for that was the fact that if you're doing something, it's always good to see what everybody else is doing. Don't just become master of that one trade. Think about somebody else. Go to lectures. You never know when you're going to hear something that will either be useful for the research you're doing at the moment, or you will decide it's much more interesting than what I'm doing at the moment, and that's what I want to go and do. And that's what happened to me with Dan Nathans. I heard about restriction enzymes. I had gone to Cold Spring Harbor expecting to sequence SV40 with using RNA technology. But I heard this lecture. It changed the course of my scientific career. Similarly, when I was a graduate student in chemistry, I read a book by John Kendrew called The Thread of Life. And that was all about the origins of molecular biology in the early days. And by the time I finished that book, I was absolutely certain I wanted to be a molecular biologist, and that's what I've become. So I encourage people to listen to things outside of their field, read outside of your field. At the moment, I'm very interested in artificial intelligence, and I'm thinking I will probably try to do something in that field at the moment, because I find it absolutely fascinating. So don't be afraid to change. Change can be a very, very good thing.
Okay, thank you. We have one more question, please. The lady on the back.
My name is Luthaila Bunsombat. I'm a lecturer from Department of Biology. I would like to say that since I was a student, when I used the resistant enzyme, the NAB always my first choice. So I would like to know if I have a DNA fragment and I would like to cut, at the specific site, but I could not find any commercial restriction enzyme. Do you have any suggestion for this? And it is possible to engineer a restriction enzyme based on the restriction site?
Okay, so there are really two answers to that question. The first, are you familiar with the rebase database? You know it? Okay, so if it's not there, then you should write to me, tell me what it is you're particularly interested in, and I'll see if I can help you to either find or make the specific enzyme that you want. We are working at Biolabs trying to figure out how to alter specificity specifically. We've not done it yet, but there are quite a few cases where we found DNA methylases that have a new specificity, but we don't yet have a restriction enzyme. And so very often that's a good place to start, because there may be a restriction enzyme that we've not recognized so far just from their sequence, but it may be in the organism anyway. So feel free to write to me. I'm usually pretty good about answering emails. Isn't that true, Uwe?
Okay, thank you. Thank you very much.
ลำดับต่อไปจะเป็นพิธีมอบของที่ระลึกนะคะให้กับท่าน Professor Roberts นะคะสำหรับของที่ระลึกเป็นกระเป๋าผ้าฝ้ายและเน็กไทพิมพ์ลายจากใบสุพรรณิกาด้วยเทคนิค Eco Printing ซึ่งเป็นผลงานจากคณะวิทยาศาสตร์โดยเป็นการพัฒนาผลิตภัณฑ์จากผ้าฝ้ายเป็นกระบวนการถ่ายโอนสีและลวดลายจากใบสุพรรณิกาซึ่งเป็นต้นไม้ประจำมหาวิทยาลัยรามคำแหงโดยใบสุพรรณิกาเป็นใบสีเหลืองทองในขณะที่ครามเป็นสีน้ำเงินซึ่งทั้งสองสีนี้เป็นสีประจำมหาวิทยาลัยรามคำแหง
Next, acting president of Ramkhamhaeng University will give a token of appreciation to the speakers. For the token of appreciations, we have cotton bags and necktie printed with Supanika leaves and indigo using an eco-printing technique. These are the research output from the Faculty of Science, Ramkhamhaeng University, developing cotton products using a dyeing technique to print colors and patterns from the leaf of Supanika or yellow silk cotton, which is the symbolic tree of Ramkhamhaeng University. The leaves give golden yellow, while indigo makes them blue. Both are the official colors of Ramkhamhaeng University.
ค่ะต้องขอเรียนเชิญท่านอธิการบดีมอบของที่ระลึกแด่ Professor Sir Richard Robert ค่ะ May I invite Assistant Professor วุฒิศักดิ์ลาภเจริญทรัพย์ Acting President of Ramkhamhaeng University to give a token of appreciation to Professor Dr. Sir Richard Roberts, please.
และก็ผมขอเรียนเชิญคุณอูเว่ มอราเวสขึ้นมาบนเวทีอีกครั้งเพื่อรับของที่ระลึกจากท่านอธิการบดีครับ And I would like to invite Mr. Uwe Morawetz again on the stage to receive a token of appreciation from the acting president of Ramkhamhaeng University.
ค่ะขอบพระคุณผู้ช่วยศาสตราจารย์วุฒิศักดิ์ลาภเจริญทรัพย์เป็นอย่างสูงค่ะ We would like to thank Assistant Professor วุฒิศักดิ์ลาภเจริญทรัพย์
ครับและกับผมนะครับขอบพระคุณผู้ช่วยศาสตราจารย์ดร.วชิรพรอาชวคมนะครับที่ได้ทำหน้าที่เป็นผู้ดำเนินรายการร่วมได้อย่างดีเยี่ยมนะครับขอบพระคุณนะครับและกระผมขอนำทุกท่านเข้าสู่การประกาศผลการประกวดโครงงานวิทยาศาสตร์ครับ
และนี่จะเป็นกิจกรรมสุดท้ายนะคะสำหรับในวันนี้กับการประกวดโครงงานสิ่งประดิษฐ์ด้านวิทยาศาสตร์เทคโนโลยีและนวัตกรรมนะคะซึ่งเป็นการสร้างสรรค์งานด้านวิทยาศาสตร์เทคโนโลยีและนวัตกรรมที่สามารถนำไปใช้ให้เกิดประโยชน์ในชีวิตประจำวันหรือสามารถนำไปต่อยอดงานวิจัยเชิงลึกได้โดยมีเป้าหมายการพัฒนาที่ยั่งยืนซึ่งจะต้องอาศัยโมเดลเศรษฐกิจใหม่ BCG โมเดลเป็นฐานค่ะ
So the science project competitions aim to encourage young students to create scientific, technological, innovative projects to support the Sustainable Development Goals and the BCG model. The project should involve the concept of BCG model, which consists of bioeconomy, circular economy, and green economy, to create solutions for daily life, and the output of the project should be the foundation for the future research.
ซึ่งในวันนี้นะคะก็มีการจัดประกวดกันใน2ประเภทด้วยกันก็คือระดับมัธยมศึกษาตอนปลายแล้วก็อาชีวศึกษานะคะซึ่งเราได้รับในส่วนของผลการตัดสินเป็นที่เรียบร้อยแล้วค่ะ
And now for the competitions, we have two levels. The first one is for the high school level, and the second one is the team for vocational school level.
เรียนเชิญท่านวิทยากรนะคะให้เกียรติมอบรางวัลให้กับน้องๆค่ะ
May I invite Professor Dr. Sir Richard John Roberts to provide the certificate for the winners.
รางวัลรองชนะเลิศอันดับที่2ประเภทมัธยมศึกษาตอนปลายนะคะในครั้งนี้มี2รางวัลด้วยกันซึ่งจะได้รับเงินทุนการศึกษา 3,000 บาทนะคะรางวัลแรกค่ะได้แก่โครงงานการศึกษาเปรียบเทียบประสิทธิภาพของสารตัวเติมจากขยะทางการเกษตรสำหรับทำเครื่องประดับศิราภรณ์จากโรงเรียนมาบอมฤตวิทยาค่ะ
The first, second runner-up will goes to Mab Amarit Vitya School. And this team will get 3,000 baht for the reward.
น้องมีตัวอย่างของเครื่องประดับศิราภรณ์มาให้ดูด้วยค่ะอาจารย์ขอแสดงความยินดีด้วยนะคะ
Congratulations.
และสำหรับรองชนะเลิศอันดับที่2อีก1รางวัลนะคะประเภทมัธยมศึกษาตอนปลายนะคะได้แก่โครงงานที่มีชื่อว่าฤทธิ์ของสารสกัดจากมะกรูดเพื่อช่วยลดปริมาณ ROS ใน Crinohabilis elegans ที่ถูกชักนำให้เกิดภาวะ oxidative stress ค่ะโดยโรงเรียนราชวินิตมัธยมค่ะ
For the second seat, for the second runner-up goes for Rajawinit Mathayom School.
ขอแสดงความยินดีกับน้องๆด้วยนะคะรางวัลรองชนะเลิศอันดับ1ประเภทมัธยมศึกษาตอนปลายซึ่งจะได้รับทุนการศึกษา 4,500 บาทได้แก่โครงงานที่มีชื่อว่าการศึกษาประสิทธิภาพเครื่องส่งสัญญาณเตือนภัยการจมน้ำในสระว่ายน้ำระบบปิดจากโรงเรียนอ่างทองปัทมโรจน์วิทยาคมค่ะ
The first runner-up it goes to Ang Thong Pathama Road Vityakom School. and they will get a reward of 4,500 baht.
ขอแสดงความยินดีกับน้องๆด้วยนะคะมากันที่รางวัลชนะเลิศประเภทมัธยมศึกษาตอนปลายนะคะซึ่งจะได้รับเงินรางวัล 6,000 บาทค่ะได้แก่โครงงานที่มีชื่อว่าการพัฒนาผนังยางธรรมชาติที่มีคุณสมบัติในการป้องกันรังสี X โดยใช้สารตัวเติมเป็นแบเรียมซัลเฟตเพื่อประยุกต์ในห้องรังสีโดยโรงเรียนวิทยาศาสตร์จุฬาภรณราชวิทยาลัยปทุมธานีค่ะ
The winner of the high school level goes for วิทยาศาสตร์จุฬาภรณราชวิทยาลัยปทุมธานี School. The winner will get a reward of 6,000 baht.
และอีกหนึ่งประเภทการประกวดค่ะเป็นของอาชีวศึกษานะคะซึ่งเตรียมตัวมาไม่ทันก็จะมีเพียงแค่1ทีมเท่านั้นนะคะที่เข้าร่วมการประกวดแล้วก็ได้รับรางวัลชนะเลิศไปเลยจริงๆเราไม่ต้องบอกก็ได้ใช่ไหมคะว่าเป็นใคร รางวัลชนะเลิศประเภทอาชีวศึกษานะคะได้แก่โครงงานที่มีชื่อว่าการศึกษาประสิทธิภาพของอุปกรณ์ดัดเหล็กจากตลับลูกปืนจากวิทยาลัยเทคนิคสมุทรปราการค่ะ
The winner for the vocational level goes for วิทยาลัยเทคนิคสมุทรปราการ Technical College, and they will get 6,000 baht for the reward. And we would like to thank Professor Dr. Richard Roberts for your kindness.
และในส่วนของกิจกรรมต่างๆในงานครบรอบ 50 ปีคณะวิทยาศาสตร์ในวันนี้นะคะก็ดำเนินมาจนถึงกิจกรรมสุดท้ายเป็นที่เรียบร้อยแล้วนะคะเราจะมีพิธีปิดงานอย่างเป็นทางการโดยท่านประธานในพิธีนะคะเรียนเชิญผู้ช่วยศาสตราจารย์วุฒิศักดิ์ลาภเจริญทรัพย์ประธานกรรมการส่งเสริมกิจการมหาวิทยาลัยกรรมการสภามหาวิทยาลัยรามคำแหงรักษาราชการแทนอธิการบดีมหาวิทยาลัยรามคำแหงให้เกียรติกล่าวปิดโครงการในวันนี้ขอกราบเรียนเชิญค่ะ
Now we are approaching the closing ceremony. We would like to invite Assistant Professor Wuttisak Lapcharan Sap, Acting President of Ramkhamhaeng University, to deliver closing speech.
First of all, I would like to thank Sir Richard John Roberts for giving us an opportunity to welcome you to Ramkhamhaeng University. Thank you indeed. And also Mr. Uwe Morawetz from International Peace Foundation. This is the second year that we met, and we hope that we will continue to meet again and again in the next future. Thus, I will speak in English, and now I was asked to come up here to make the closing remarks. Since this morning I speak in Thai, so I will continue my closing remark in Thai. If I may apologize, okay? ก็ขอบคุณทุกท่านนะครับที่เรามาถึงโอกาสสุดท้ายที่เป็นการปิดโครงการเสวนาแล้วก็เป็นการเสวนาทางวิชาการทางด้านวิทยาศาสตร์ซึ่งในครั้งนี้เนี่ยเราได้รับโอกาสที่ดียิ่งจากเซอร์ริชาร์ดจอห์นโรเบิร์ตส์ซึ่งท่านได้อุตส่าห์เดินทางมาแล้วก็มามีส่วนร่วมในงานในครั้งนี้นะครับต้องขอแสดงความยินดีกับลูกๆทุกคนที่ได้รับรางวัลแต่สำหรับโรงเรียนที่ไม่ได้รับรางวัลก็อย่าเสียใจนะครับความล้มเหลวก็คือสิ่งที่จะทำให้เรามีพลังที่จะทำให้เราคิดต่อไปว่าในโอกาสหน้านั้นเนี่ยเราจะทำโครงการอะไรบ้างนะครับสำหรับคณะวิทยาศาสตร์ของมหาวิทยาลัยรามคำแหงนั้นเราจะ ส่งเสริมโครงการนักศึกษาเช่นนี้ไปเรื่อยๆนะครับคุณครูที่มาเป็นผู้ดูแลลูกศิษย์ลูกหาก็อยากให้คอยติดตามข่าวสารจากทางมหาวิทยาลัยหากมีโครงการอะไรก็ตามที่เราสามารถที่จะช่วยสนับสนุนหรือทำให้ได้เนี่ยก็ขอให้บอกนะครับวิทยาศาสตร์ของเราคณะของเรานั้นเนี่ยครบรอบ50 ปีวันในครั้งนี้ก็เป็นโอกาสที่ดีที่เราได้มีการจัดงานขึ้นนะครับแล้วต้องขอบคุณทุกท่านกรรมการทุกคนท่านคณบดีคณะวิทยาศาสตร์นะครับและกรรมการที่จัดงานตลอดจนคณาจารย์ที่มาให้กำลังใจ ลูกศิษย์ลูกหาในวันนี้นะครับขอบพระคุณและขอปิดการประชุมครับขอบพระคุณครับ
กราบขอบพระคุณท่านผู้ช่วยศาสตราจารย์วุฒิศักดิ์ลาภเจริญทรัพย์ประธานในพิธีในวันนี้นะคะขอบพระคุณวิทยากรทุกท่านนะคะที่ทำให้กิจกรรมในวันนี้นะคะสมบูรณ์เป็นที่เรียบร้อยรวมถึงน้องๆนะคะที่เข้ามาร่วมประกวดในโครงงานต่างๆวันนี้ด้วยแล้วก็กราบขอบพระคุณแขกผู้มีเกียรติทุกท่านที่ร่วมกิจกรรมในวันนี้ด้วยค่ะ
We would like to thank Assistant Professor Wuttisak Lap Charan Sap for your closing speech. And on behalf of Ramkhamhaeng University, we would like to extend our heartfelt thanks to all of distinguished and honored guests, speakers, and any participants joining these events. We would like to also express our sincere thanks to students, youth, and teachers joining these occasions too. And we wish you all safe journey home, and we hope to see you again soon.
ค่ะขอบพระคุณทุกท่านมากนะคะแล้วก็หวังว่าจะได้รับความร่วมมือจากทุกท่านอีกเวลาที่เราจัดกิจกรรมแบบนี้นะคะวันนี้นะคะดิฉันสุภาวินีบัวบานจากสำนักเทคโนโลยีการศึกษาค่ะ
และผมนายวิชินสืบปราจากภาควิชาชีววิทยาคณะวิทยาศาสตร์มหาวิทยาลัยรามคำแหงครับ
ขอบพระคุณทุกท่านและสวัสดีค่ะ
สวัสดีครับ